Hello, everybody. This is Nirja Sankaran here to welcome you back to the DNA Papers, a podcast series that aims to bring home the science of DNA by unpacking and illuminating the original research that went into the discovery of the substance and understanding its role in life. After detours in our previous two episodes to cellular genetics and microbiology, where DNA was scarcely mentioned, we return here in our fifth episode, and for the rest of the series, we'll be talking about the material, even if it isn't quite called DNA yet. Those following the series may remember that in episode 2, we discussed the contributions of Albert Kossel, who in 1910 received the Nobel Prize in Physiology or Medicine for his discovery of the different nucleotide bases, A, T, G, and C, that make up the DNA molecule. Today's session will focus on the work of one of the many scientists mentored at one time or another in their career by Kossel. The person is Phoebus Levene, who, among other things, discovered the sugar components of the nucleic acids, the ribose in RNA and the deoxyribose of DNA. I have a quick word of explanation before I introduce our guests and leave the conversation in their capable charge. Today's episode deviates in many ways from the pattern established in our previous conversations. Most significantly, it is not about one or a few of Levine's papers on DNA per se. One reason for this is that Levine was amazingly prolific and over half a century produced hundreds of papers in both German and English, titled in variations of the theme on the nucleic acids, and it would be difficult to privilege some over the others and offer a fair assessment of his work. More intriguingly, however, not one of these papers contains the actual elaboration of the idea that Levine is probably best remembered or misremembered for today, something called the tetranucleotide hypothesis, often blamed for obstructing the realization of DNA's importance to life for decades. So, paradoxically, and what would history of science be without its paradoxes? Today's episode is about a paper that never was. Rather than reveal anything more, I'll turn to introducing our guests. First up, I'd like to welcome back historian of science Panina Abiram, whose first history of science publication in 1978 was, as I mentioned last time, a review of A Century of DNA, one of the sources of inspiration for this entire series. The book was one of the first books to give Levine a fair representation in the DNA story, which is a fact that Panina made a special note of in her review. I'm very grateful to have her return to the series. Thank you for joining us, Panina.
Thank you.
Our next guest is Pedro Bernal, a professor of chemistry from Rollins College, Florida. Pedro developed a deep interest in the history and philosophy of science during his undergraduate years at the University of Tennessee in Knoxville, where he also obtained his PhD in chemistry. Whenever and wherever possible, he attempts to incorporate these interests into his teaching and in writings about the value of history and philosophy of science in or for science education. Welcome, Pedro. I'm very pleased that you're joining us.
Thanks for having me.
Also, returning to this series to pick up the threads of our previous conversation on DNA chemistry is Mark Lorch, who's a working biochemist as well as a communicator of science at the University of Hull. I'm especially keen to hear his interpretation of Levine's misunderstood stance on the much maligned tetranucleotide hypothesis. Welcome back.
Great to be here. Thank you very much.
Well, welcome again, everyone, and thank you for joining this conversation on DNA chemistry. Now, usually I begin each podcast session by asking the guests to offer some detail about the content of the papers under discussion. But as I said, this episode does not focus on one or a few papers. So I'm going to open with a slightly different opening gambit. Why would you say that Levine is deserving of his own episode in a history of DNA? Panina, would you like to go first?
Sure. He deserves a special episode because for 3 or 4 decades, ending with his death in 1940. Certainly since 1909, he and his lab were the main workers on DNA chemistry and the chemistry of its components. And this period, which includes the entire interwar period as well as time before World War One, is a very crucial period in the history of science in the 20th century. Whether we count from 1905, when he got the jobs that enabled him to do what he did, or 1910, we talk about 3 decades of being at the very center of, we should say, chemistry of natural products. And biochemistry less, because his enzymatic work was very limited. But in terms of chemistry of natural products, which is the most complicated chemistry that exists, and those who majored in chemistry know that you study it only after you finish organic chemistry, analytical chemistry, and all the type of chemistry that exists, because it is so complicated. And indeed, the nucleic acids, of which DNA and RNA are the representatives in this session, are very complicated chemically, and to resolve their structure is the major achievement of Levine and his lab.
Would Mark and Pedro like to add anything?
Yeah, I'd love to. So I think the thing about Levine is and why he deserves his own episode is just his. He was so prolific, and so dedicated to the cause. He was held up as this, champion of nucleic acids. He published 700 papers throughout his career. He he worked at the Rockefeller Institute on right up to the day before he died. In at the age of 71. So is just such a vast contribution to the field, and absolutely you can't have a story of DNA without mentioning him. He defined so much of the chemistry, or discovered and published so much of the chemistry that we're now so familiar with.
May I add a point, please?
Sure.
He was also important institutionally, which, there was a time when historians of science were only interested in ideas. But since then, we are also interested in institutions. And the fact that he was among the founding members of the Rockefeller Institute, now Rockefeller University, means not only that he had a very good job, but de facto he had unlimited resources. So I would say that whatever he and his lab achieved was the best that could have been achieved worldwide, because I don't know of a better endowed institution in that area at that time. Only Lister in London and Pasteur in Paris were comparable. And I still think Rockefeller had more resources. But even if we compare with the other two, and the other two are more focused on infectious diseases and stuff like that, but complete freedom for basic science was very rare at the time, and he had unlimited resources. Even Simon Flexner, the director, if he wanted to cut his budget, Levine put up resistance and the budget remained. So this is really a unique situation that all of us only dream of and very few people achieve. So it's the top of the line in institutional terms.
Great. Thank you for that bit of information, Panina. Pedro, would you like to add anything at this point?
Yes, I think Panina points to something that's really important. If one looks at the descriptions of Levine, he might be referred to as Russian American biochemist. I'm not sure that that's the best description for Levine. Levine, on my reading, was basically someone that thought of himself as a traditional organic chemist, and he was incredibly good at it. The achievements of Levine in figuring out the structures of the sugars and the publication of the 1935 structure of the nucleotides, etc., is just a major achievement for anybody, for any career. It's an amazing achievement. I'm not quite sure, to be honest, what is the reason for the fact that he's remembered presumably for something he got wrong. In my reading, I just don't see it. Everything that he did, essentially concluding with the structure that was published in 1935, qualifies him as undoubtedly the preeminent chemist on nucleic acids, even though, and I'm taking my cues here from Hunter, who wrote a long article on him, only about 25% of Levene's papers were referring to nucleic acid. So there is a huge amount of work that he did that sort of never gets talked about a great deal. But in addition to his work in nucleic acids, he just did amazing chemistry in other areas as well.
May I, Nirja, point to a similarity and dissimilarity with another scientist? Because after all, we all try to solve the puzzle at the center of this session, which is why, like Pedro said, and others, Grammy Hunter and others, if he wrote 700 papers and did and solved the chemical structure of DNA and RNA, these are tremendous achievements for anybody. Why the only thing that people want to remember is that due to ambiguous formulation, some people thought that he had the wrong ideas, that he disqualified DNA from being the genetic material, which is a very big disaster, like one paper said, a scientific catastrophe. So I want to just make a brief comparison with Linus Pauling, whom you all know and must know. Linus Pauling solved 225 structures, and this is probably more than any other chemist. Some of them were amazing, like alpha helix of proteins are major, major achievements. And others are also important achievements, but of a more regular manner. But the great Linus made a big flop with DNA structure by claiming that there are 3 strands, also that not being an acid but a salt, and all kinds of mistakes that it's impossible to clarify. But nobody says that Linus Pauling is remembered only for his paper of DNA, which is a disaster indeed. And nobody can understand how the best chemist or one of the best makes elementary mistakes that even 2 incompetents like Watson and Crick could spot. So there are incomprehensible things, but what should really focus us is why this type of bizarre charge seems to stick in Levene's case and doesn't stick in Pauling's case.
I think the answer partly there is, is that, of course, Pauling's paper on describe the DNA triple helix, etc., with the bases pointing out, as you say, absolute disaster. The amazing actually thing about that always amazed me is that he completely ignored hydrogen bonding, which was, of course, the thing that he described. So but anyway, we're not talking about Pauling, are we? We're talking about
there is nothing there that is not wrong.
No, exactly. Yeah, great. That's a perfect way of putting it. So, but I think the difference is, of course, is that with Pauling, that paper was very quickly corrected, and it didn't, by Watson and Crick and so on. And whereas Levene's is accused of holding back DNA research by fundamentally, not necessarily because of the tetranucleotide hypothesis, but because he didn't believe that DNA was the molecule of inheritance, and that, of course, went on for so much longer, isn't it? I think if Pauling's structure had persisted for a long time and had become the dogma, we would have a very different view of Pauling. But yeah, I think that's the just fundamental difference, isn't it? Is how long the actually Levine supported the dogma that DNA and proteins were, and but DNA wasn't the molecule of inheritance, and it was proteins that were responsible.
Plus, if I may interject, going back to what I said in the beginning, there was a real paper, I mean, there was an actual paper from Pauling where he has all these spectacular mistakes, as you call them. So one could start to disprove it. The thing with the tetranucleotide hypothesis is that it sort of came into being as something, like Minerva. It's just there, and it's attributed, the fact that there are these repeating units of all four nucleotides is just attributed in retrospect almost to Levine. When Levine mentions a tetranucleotide hypothesis himself in one of his papers, it really doesn't say that, does it? That there are these four repeating units.
Because they didn't know at that time. They didn't have evidence for the order of in polymers. They didn't realize at that time most polymers had the same monomer, so it didn't matter the order. That could be a polymer that the order is encoded. Nobody could think about it at that time. And even for proteins, the order of amino acids was discovered only, I think, in the 50s, which is relatively late. Before, they didn't know that the order in a protein matters. It's not just the composition. And chemistry was primarily about composition and not about order. And, to blame somebody for something that was not done at that time, it's
That's right. I mean,
not very helpful.
I agree. I mean, you look back through some of those papers, and it's really difficult. When I started looking back again, I was like, well, where did this spring from? This tetranucleotide hypothesis, and you can see it sort of almost evolving from a diagram here, which shows all the four nucleotides together, and another diagram, there, and then the scientific community must have just sort of slowly come around to this idea. And I suppose actually when you look at it, it doesn't look unreasonable. From their analytical data, they look like there are these four bases in equal amounts. And so if you apply Occam's razor, that the simplest hypothesis is probably true, it's not an unreasonable conclusion to come to. Some of the more weirder things that emerge, like there's the ring diagram, which shows the four nucleotides, looped together in a ring. Where did that actually come from? I may have overlooked the paper in there or couldn't find it, but maybe Pedro or can you shed some light on that?
I've never seen the actual paper where the circular structure is. The linear structures, Hunter's article actually showed them in some detail, ending up with a 1935 structure, which is essentially correct. I've seen from Hunter's paper and others sort of the development of the structures that were published, I think starting in 1912 and concluding in 1935. And they, of course, are sort of improvements responding to the chemical evidence that he's getting. So Penina talks about Pauling, but Pauling comes from a whole different tradition, right? Where we now have the benefit of being able to do sort of structures using X-rays and so on, and that's in the middle of the work among proteins and so on. Levine has essentially zero access to that kind of data. By the time it comes around, he's been dead a few years. And this is sort of just pure chemistry and putting things together based on the evidence that just comes from the way these things react through hydrolysis and so on. To be honest with you, it seems to me sort of almost, a work of genius to be able to get to that kind of detail from just evidence that doesn't have a structural component. On the other hand, keep in mind that the information on the molecular weight just begins to come available when Levine passes away. So he wasn't really talking about polymers because other people were sort of battling it out, right? Stodinger's and others, about whether you had what we now call polymers, so you actually had colloids, which were basically a bunch of monomers together. We now know that you can have both at the same time, so that's not like one or the other. But toward the end of his life, evidence on large molecular weight started to come in, and he didn't have any problem extending this tetranucleotide into a much larger polymer that was going to accommodate that. So it doesn't seem to me that he was wedded very much to the, so just the tetranucleotide as a claim that that's the way things were, but just the best he could come up with based on the chemical evidence that he had. And as I said, the 1935 structure, essentially just a single strand, is pretty much the correct. I don't know what evidence he could have had to basically show him that what he was proposing was incorrect. So I will not call it a mistake.
Great. I want to back up a little bit. Some of the things you've said, all of you, sort of beg some questions, and we need to back up. First of all, could one or all of you offer some background into Levine's education and training? How did he develop what one author about the DNA story has called, and I quote, a virus like passion for the fantastic nucleic acids? So, evidently, he thought nucleic acids were fantastic. Could one of you please elaborate on that?
Well, what I can say is that he had a very privileged education. We know that in the Russian Empire, Jews were not allowed to live except in the Pale of the Settlement, which corresponds to current day Belarus and Ukraine. And to go to the Imperial Military Academy in Leningrad was not the average trajectory for people of Jewish background. Somebody in his family must have been of some great importance to be allowed that type of education. His teacher in chemistry was the most famous chemist in Russia at the time, Alexander Borodin, and he developed first a passion for chemistry, and now whether nucleic acids were, they seem to be an addition from his time in Kossel's lab, because he works on lipids and he works on proteins and he works on almost every chemical compound. But Hunter says this quite nicely, that he paid attention to the fact that nucleic acids, together with in the form of nucleoproteins, are always involved in some key biological processes like generation, regeneration, which were of interest at the time. So he was not totally unaware of the biological importance, but what seems to be a puzzle is that he did not seem to maintain a contact with people who are working on cell chemistry and would have come to DNA's biological role by other routes, especially since someone like Avery was in the same place. Hunter says that in the last 23 years of his life, he looked at nucleic acids as a chemical challenge, not as a biochemical or biological. Fair enough. I mean, he was a chemist, a very great chemist, a very sophisticated, and you cannot solve biological problem with chemical tools. But this is what I find puzzling, is that despite being situated at the very center of the American biological and chemical world, he didn't hear, or there was nothing to hear, by the way, because biologists were not zooming on DNA's role. Everybody thought it was midwife for proteins. So he didn't miss anything. Biologists, nobody was saying that DNA was alone of importance. So to blame him, it's again bizarre, exceedingly so.
I suppose, though, I mean, he was the world leader in nucleic acids, so people are going to follow his opinion, and when he makes comments about nucleic acids carrying no individuality or specificity, and that they could not determine species specificity or be the carriers of Mendelian characters. He's saying that, and the rest of the field's going to follow. So I think whilst we can't blame him for this tetranucleotide hypothesis that everybody frequently does blame him for, I think he does have to take some element of the blame for holding up DNA research so long by simply dismissing the idea.
But nobody forces other people to challenge. If there were other major labs in Germany and in other countries that could have challenged, if anybody thought that nucleic acids were really important, as it happened indeed in the late 40s and early 50s. People realized that these are not the midwives. They are important in their own right, and they did what was done to give them the standing. Nobody thought about it, and they were not blocked by Levine. They're blocked by their own zeitgeist to which they subscribe.
Well, I mean, but he was part of that zeitgeist, wasn't he? He was a
Yes,
driver for it.
yes, of
Yeah,
course.
yeah, yeah.
No, if possible, just to say a little bit more about the educational track, right? So he came to the U.S. when he was 22 years old. He had already gone through his medical training, but he went back to his medical studies in Russia, and after finishing, he came to the U.S. and found started to actually practice medicine while studying chemistry at Columbia, and he found a job in a pathology lab in the state of New York, and that I think is where he first encounters nucleic acids, and then he basically gets tuberculosis, and like Walker Percy spends 4 years sort of going back and forth, and he seemed to have changed his life in sense of direction, because it's through that connection with tuberculosis that he during this period he goes back to Europe and works in the labs that have been mentioned, including Fisher, which is of course central to his sugar work. And then when he comes back to the States, goes back to Saranac, I think is the name, which is tuberculosis, a place where people could recover, but there was a lab associated with it. And it is from there then that he goes to Rockefeller, in 1905, and I think in 1907 then gets appointed as the chief of the chemical division, and he spends the rest of his life there.
Right. So, how did he get interested? Where did he first get into nucleic acids? Could anybody answer that question?
Yeah, I think Hunter makes a reference to that he was in the pathology lab that he first saw them, started to work with them.
And when did he work with Fossil?
The visit, like Nirja says, the visit in Russell's lab must have been there something that attracted his attention, because another thing that could be he was what used to be called a cultivated scientist, as opposed to a mere technocrat or robot or whatever. He was very, by cultivated, I mean that he knew several languages, he read literature, theater, stuff like that. So he must have read around, and something must have captured his interest, because Hunter says very clearly that it was his encounter with nucleic acids in connection with regeneration, biological regenerations, that this is a biological riddle of long standing, why certain organs regenerate and others don't. And to solve such a problem would have been a very big deal. So he had a motivation that was biological, but the tools that were required, the chemical detour was very long, and apparently he never had the time to go beyond it, or the opportunity.
If I could say a few words about Mark's point on how influential he was in terms of sort of keeping people away from nucleic acids, it might have been the issue of the separation of proteins from nucleic acids, so that one could isolate what is it that's doing the job, right? Was a huge complicated problem, and my sense is, even though this might be an entire different program. My sense was that the vast majority of people within the biological community, from which Levene was probably a little far removed, to be honest, were of the impression that proteins were the carriers of what later on becomes known as a transforming principle, right? And you can see people years after the structure of DNA was revealed, still talking about the fact that we're not sure whether we got this stuff clean enough so that we can make that claim. And so part of there's a sense in which when we talk about histories, of course, the fact that you figure why what's Levine spending 30 years on, or what's Avery spending 30 years to figure out, is that these processes that requires for you to actually clean these molecules are basically what takes years from day to day, the work of the professors and the graduate students mostly, right? And it's something that it basically tends to disappear in the background as we talk about this sort of theoretical stuff that shows up in the papers, right? Most of these things don't show up in the paper with the level of detail that they merit, because you probably have 2 PhDs on cleaning up a molecule,
And that's a really interesting point you're saying about the papers not having the level of detail, because I mean that's actually one thing he was notorious for, wasn't it? Just not including enough detail in his papers. You look at so many of them and they describe some of the procedures as being carried out in the usual way, which is the sort of thing that I'd haul my students over the coals for, writing up a method in using terminology like that. So, and I think again, the review written by Hunter describes how he published a series of papers going from 1 to 11, all with the same titles, but just numbered 1 to 11. But he missed out 9 and 10. So he clearly sometimes didn't have quite the attention to some detail that we would really prefer these days, certainly.
But what Pedro said, and it's very important, and this gives us, or at least it gives me, a new angle on Levine. Everybody believed that in nucleoproteins, which were associated with heredity in one form or another, proteins were the main players. Everybody believed that. Now, to be a Levine or an Avery means to be unusual, because you deviate here from the consensus that proteins are important, which means that he was rebel of some sort. And you go and work on something that everybody thinks it's less important. Not everybody does it. Most people would go, what was the joke in the lab? Go find yourself a good protein, which means easy to crystallize, to get to get some diffraction lines. This is what people were told to do, not to look even in the Cambridge lab. These are the. So if we look from this angle, it's possible that Levine thought that he was going to do something that nobody gambles on. Everybody gambled on proteins. To do nucleic acids is really a dissident position, intellectually and technically. And we haven't said it yet, but he was technically very astute, and every innovation that was available in structural chemistry, be sure he applied it to the subject.
Yeah, and since we all now work with molecules of biological importance, it's sort of difficult to imagine the attitude of chemists to biological molecules at the time, right? Because no serious chemist would work with these things which are essentially impossible to isolate in a way that you can do any decent chemistry on. So if one looks at the controversy between polymers and colloids and so on, basically what Staudinger gets told over and over again is, look, if you want to do any serious chemistry, forget all these big molecules, because you cannot get them in a state where you can do any chemistry that will tell you anything, right? We now look back that, okay, without polymers, what is there to do? But at the time, if you needed to concentrate on a small molecule so that you could do your chemistry and claim that you knew what you were working with.
A quick interjection question here, please. Pedro, could you just give, for the sake of our audience who may not be quite up to date on all the names, Stadinger, what was his full name and what was he best known for?
So Hermann Staudinger was a German chemist that eventually won a long-running controversy about the nature of large molecules, macromolecules, where they
Sorry, and this was in the 20th century or late 19th century?
yes, this is at the beginning of the 20th century. Staudinger got a Nobel Prize for it in, I think, yes, the 1940s and so on.
52.
So Staudinger is still alive when the polymer industry begins to take off, and is now, of course, the at the time, of course, the structure of plastics and rubbers and macromolecules of all kinds was in dispute, and basically the argument was: are these sort of agglomerations of very small units, or were they sort of continuous strings? Within the conversation that we're having, we're talking about tetranucleotides, right? Okay, so do you have this repeating itself in a linear chain that forms a large molecules, or do you have very small components, which actually Castle was, I don't remember the German term for it, but it's basically the building blocks hypothesis, if you will. Do you have these very small parts agglomerating themselves? And that's why it gives you the impression that they are much larger molecules. And of course, the history of physical chemistry has a period at the end of the 19th century and the beginning of the 20th century, which is essentially dominated by the chemistry of colloids, where people thought that they were always talking about this conglomeration of small molecules, when in fact some of them were basically sort of a linear repetitive string, so what we now call monomers, right?
Absolutely. Thank you so much. Two other points I wish to clarify. One, I'd just like to point out to the reader that the hunter that is referred to is a scientist who's written who's done historical work on the history of DNA and has written a paper that is in our further resources list about Phoebus Levene specifically and this tetranucleotide hypothesis. The second clarification before I get to that, Mark, you have something to say.
I was just going to say I'm aware we still haven't really described what chemistry Levene did. We've talked about what a great contributions he's made, but of course, what did he do? And I mean, if you don't mind, I'll go first with that. Essentially, he identified the sugars involved in the ribose and eventually deoxyribose in DNA. He worked out the way that the phosphates, the sugars, the bases were all linked together, and he coined the term the nucleotide. So these are the fundamental building blocks of DNA, and this tetranucleotide hypothesis that we've mentioned, and that we are mentioning, is essentially comes about because the amount of the four bases in DNA, A, T, G, and C, is certainly in the organisms he was looking at them in were more or less equal, at least as far as his analytical techniques could tell. Therefore, it was reasonable to assume that the basic building block was formed from these four nucleotides being joined together to form some sort of monomer that might be then repeated on a larger scale. So and we've talked about how long he worked in this field for, the whole of his life, for years and years. And I suppose to our mind now, looking back, you think, well, it took somebody that long to work up to find these solutions, but it was incredibly challenging, as Pedro said, purifying the amounts of DNA, finding the right conditions to break up these components, hydrolyze these various components, and then analyze them. And I think, I mean, maybe what one of the other guests can shed some light on this, but there's this wonderful description in Gareth Williams' book about unraveling the double helix, where he describes how one of the breakthroughs was finding the right conditions to break down thymus DNA, yeah, DNA from thymus, and he described using an incubator that was basically a dog's intestine, a living dog's intestine. Uh, and I mean, I haven't found any description of it elsewhere, but so maybe one of my maybe somebody else could shed some light on that.
I just wanted to ask you to clarify one thing, please. We talked about the fact that A, T, G, and C, the bases, in a previous episode, that Kossel is who named them. And now you mentioned that
Yeah,
Levene names the nucleotides. Now, oftentimes, I think as a shorthand, people think of AT and G and C, AT, G, and C to be the nucleotides themselves. Could you clarify the difference or the exact relationship between A, T, G, and C and the nucleotides? Are they part of one another, or how does it all fit together?
okay, yeah. So a nucleotide is comprised of the phosphates and the sugar that forms the backbone of the DNA, and the variable part, then the bit that defines whether it's A, T, G, and C, is the base. So you've got the four different bases in DNA, and each nucleotide then has the same phosphates and sugar and a different base. And the difference then is, of course, between RNA and DNA is also in the sugar. So in RNA, you've got a ribose sugar, and then you've got the A, G, C, and U for uracil instead of thymine for the T. So summary, yeah, it's the base changes, the sugar and the phosphate stays the same in the nucleic acid that you're interested in.
And it was Levine that put all of this together, these different pieces of the puzzle, and gave the name nucleotide.
Yeah, that is correct.
The bases are just one or two rings, depending whether they are a purine or a pyrimidine. And the latter have one ring, the former have two rings. And there are two bases with two rings and two bases with one ring. And therefore, the number of hydrogen bridges are different but these are just nitrogenous bases. They are not nucleotide requires, like Mark said, three components, and he clarified how the bases are on which C of the sugar they are connected, how they are connected. This is a very big, they gave the Nobel Prize to Alexander Todd for clarifying where the connection is on the sugar, which is a 3 and 5. But this is essential. Most of this work was done by Levine's lab and by somebody in Todd's lab. And it's not the only time when the wrong people get the Nobel Prize.
Yeah, I mean, and you can look back through the papers and you see this evolution of Levine's ideas about how the bases are connected to the sugars and the phosphates. You know, there's some really crazy ideas actually in there, but that's okay. Well, crazy from our point of view now. That's okay, but you see this evolution of him figuring out what the sugars are, then and then coming up with different hypotheses for how the sugars and the phosphates and the bases are all linked together. And of course, the other big thing that was going on at the time was whether actually the sugar was the same in plants and animals.
Yeah, right.
So Levine identified the five-membered ring, the five ribose sugar ring in from yeast, but his great rival, and they had a wonderfully competitive relationship, Walter Jones, thought that that from animals it was a six-membered ring, and it took something like 15 years for Levine to eventually show that it was a five-membered ring of ribose in all the nucleic acids, and that's where he again I came across this description of him finally finding the correct way to break down the thymus DNA and using, you say, a live dog intestine to do so. And again, back to that, because I'm fascinated by that, because I couldn't find anything else about it. So I'd love to know whether anybody else has come across any descriptions of that as well.
Pedro, you had something on this?
Just to add that, just for the benefit of the listeners, part of the difficulty here is that once we think that we have the base, the sugar, and the phosphate, we essentially have the problem solved. But the number of ways in which these things can be bonded, to say nothing of how they can have three-dimensional structure, is an incredibly difficult problem. So the road from identifying the chemical structure of the three components to a proposal that actually has correct ordering of them is a huge step, right? Because just sugar chemistry by itself, it's incredibly complex because sugars are molecules that have all kinds of different structure, pyranose, furanose, all kinds of different arrangements. Yes, you are working with chemicals that by themselves are really difficult to characterize. And so to come up with how they are bonded to each other is a major challenge, even after one knows the molecular formula for them.
Okay, thank you. I did want to go back and ask. I mean, we did get into a little bit of what exactly the tetranucleotide hypothesis or theory was. I'll repeat that. It might be because we've talked a lot about it, it might be worth clarifying it a little bit for our audience. When mentioned in texts by others, it simply describes the idea that nucleic acids exist as represent repeating groups of four nucleotides, and this was reinforced by a 1909 publication by Levene. Now, you all have already made it abundantly clear what a complex task and how long it took Levene to work through all the nucleic acids. Could you comment on what, in your opinion, has caused this diagram and the statements about the tetranucleotide hypothesis to be misunderstood and to be judged so harshly in history, fairly or unfairly?
I think that all the harsh judgment goes back primarily to Bentley Glass, who was a member of the genetics establishment. I don't think he's known as a great geneticist, but he held very important positions in genetical societies and journals, and he felt Bentley Glass.
Yeah, when? Which period?
I would say between the 1940 and 60, but probably more than maybe after 62. But he was in the transition period that we talk about in the 40s, and he felt that he was the establishment in a way. And what happened is DNA becoming alone the genetic material was very embarrassing, not necessarily to chemists because they never professed to study heredity, but to biologists. So people in certain cultural traditions, they know what to do best is to blame someone else, not from the group. And he came with this term scientific catastrophe, and this is totally absurd. He was no historian. He did not study what Levine did. He had no clue. He was just looking for an easy scapegoat. What is interesting is not so much why Bentley Glass did it, because I can see this very easily, but why more intelligent people were tending to repeat it in passing. Nobody analyzed, because once you start to look at Levine, you realize that all the statements are ambiguous. It's not easy to pin down. So I assume that the genetic establishment per se was looking really to save his skin. And the best way usually is to find somebody else to blame. And this is what happened here. I don't see any other more interesting way to solve this.
So, in other words, you're saying that The tetranucleotide just became a scapegoat to blame the delay for accepting DNA as the genetic molecule.
Uh, But it was not a question of delay. Everybody knew that nucleic acids are part of nucleoproteins, and this is a component that is involved in heredity, in cytogenetics, whatever, chromosomes, whatever you have. But nobody thought that this is the main component or the only component. And there was the block, not what happened in chemistry. This is, or what was the structure. These people didn't know the structure, even if you bang a nucleotide on their head. As it is, because it's complicated to show that the linkage of the backbone is only at carbon 3 and 5 of the pentagon. It's very hard to do that. This is why Todd got the Nobel Prize.
That came much later. That came later than Levine. But thank you. I'd like to back up and ask Mark and Pedro whether you have anything to add to what Panina said about the notoriety of the tetranucleotide hypothesis.
Right. Yeah. In addition to what Panina said, of course, that there is a tendency, not necessarily among scholars, but in terms of the way that stories about the history of science are done in the press, and sometimes, to be honest, with people that ought to know better, like Bentley Glass, right? Which tends to minimize the achievements of other people and to turn them into, how should I put it, to decrease the importance or to uh, blame some scientific development having delayed something else. These kinds of things can only be done looking backwards, and for some reason, there is part of the community that comments on science that seems to be interested in painting a picture that tends to sort of minimize the achievement that people that have come before. Not only does it apply to people, sometimes it applies to theories, sometimes it's oversimplifications about the difficulty of a particular development at a given time. I can favorite two examples are phlogiston, for example. So phlogiston was supposed to be this joke, but a joke that lasted for 130 years and nobody seemed to get it. Any hypothesis that supposes that for 130 years everybody was dumber than you is probably wrong. So obviously people were looking at something that looked like it was convincing, and for us it's very easy. And the way this story is told is that here come Lavoisier and phlogiston goes out the window. But when you look at the details, you see phlogiston sort of hanging on years and years after Lavoisier. So the history of science shows you all the time, over and over again, that whenever you have something that's supposed to be a crucial experiment, is normally a crucial experiment that only becomes crucial in retrospect, because at the time it was simply a development. And of course, the evidence begins to sort of come together into a picture. And the word discovery is sometimes sort of makes that worse, because a scientific discovery is not like discovering you have a flat tire. A scientific discovery is a process that sometimes takes years and years and years. The history of science is complicated, and the tendency to sort of be simplistic about what is going on here is just not helpful. And it tends to give people the impression that science is a very different endeavor from what it is. Responsible for the fact that those of us that are scientists are supposed to be some kind of different species.
I think that's a great point, actually. And I think it's something that we should we as scientists, science communicators, science historians should make it really spend more time making it clear that research in science is often goes up blind alleys. There's a great quote: "Research is the process of going up alleys to see if they are blind." It was first expressed by the zoologist Marston Bates. And I think that describes it perfectly, isn't it? You've got to explore. You're mapping out the possibility landscape, and sometimes that's going to involve going down blind alleys, and those blind alleys can be really long. in this case where DNA was put aside as a relatively unimportant molecule for such a long time. But that's how science works. And go back to actually Pedro's comment about phlogiston. Actually, I just to say that I wasn't aware that it was a joke, but it reminds me of maybe that was the first example of Poe's law. I don't know if you've come across that. Poe's law is the adage that in the internet culture, anything without saying that without a clear indication of the author's intent, any parody or extreme view can be mistaken by some readers as a sincere expression of their views. So I think maybe the phlogiston was the first example of that, well before the internet age, anyway, as well. But anyway, I digress.
I mean, thank you all very much. I think Pedro's point that Mark reinforced about the blind alleys in science and that science can take a long time and how historians of science need to pay attention to this is a very great point at which to end this very rousing conversation, for which I'd like to thank all three of you for participating.
Thank you.
Thank you.
Thanks. It was great to be here.